Okay, so genetic modification, part two. Last time we discussed human nature. Today, we're going to discuss the history of genetic modification. This will take us throughout several decades. We're going to cover quite a bit of stuff, so strap in. Shotgun sequencing, I think if you're a scientist, a microbiologist, or anybody that deals with genetic material, you'll understand that shotgun sequencing was a way of speeding that up, and it was a big breakthrough. And if you're not, just know that there was a big breakthrough. In 1978, 42 years ago, Louise Brown was born in England. At the time, it was one of the most controversial events, right? There were her parents spent a lot of money and they traveled to a lot of places to be able to have children. They couldn't have children, and so there was a big uproar at the time to see. People thought that it was going to spell the downfall of society, and in fact, there were various religious groups that argued that it was unnatural, that it would pressure women to have children, or that maybe even the babies themselves would be ostracized, like cultural issues, right? So religious issues, cultural issues. The Pope at the time expressed concerns that women would be used as baby factories if they could have children for other women. People thought that it would end the nuclear family. And people worried that there were going to be biological issues with the babies, they'd be deformed or dead. Okay, so when trying to analyze this with some 40 years of hindsight, to address a couple of these questions, I don't know what it means to say something is unnatural. And I don't know why it matters. Lots of things are unnatural, and naturalness can be understood in a lot of different ways. It's not obvious to me that something being unnatural makes it bad. There's got to be a separate argument for why it matters that it's unnatural. Society will pressure women. Society will shun IVF babies. Yeah, I think some people view women not having children as something bad that happened to them. It's not a majority, but it's still some stigma, and especially when you talk to people that have gone through IVF, they tell you that they suffered that stigma and they wanted to have children because of that. It's not a lot of stats on how people think about IVF offspring. I know some IVF people, I don't think very differently about them. The Pope's worry that the IVF would create an industry of professional mothers, yeah. Weirdly, that panned out. There's entire countries that have had to specifically outlaw medical tourism because American couples were going there and paying $10, $20, $80,000 in order to have somebody else carry their child to term for them so that they didn't have to go through pregnancy. And that person that would carry the child to term would receive a fraction of that. It was a life-changing amount for them, but not life-changing enough to take on serious risks unless they were extremely poor, which usually these people were extremely poor, right? These were people in Mexico or parts of India. And so countries have been playing whack-a-mole with this to try to stop European and American and Chinese tourists from coming in and having their offspring be born from somebody in that country. There's a video you can click through in the PowerPoint. Families will end. In vitro fertilization costs tens of thousands of dollars. And even then, it gives you like a 25% chance of success, depending on where you go. I don't think that is, at least in the near future, going to end just, randomly, drunkenly hooking up and ending up pregnant. Um, Claim babies will be deformed or dead. Actually, there are some anecdotal data that IVF offspring have health issues, but by and large, there's not very much of a difference. IVF technology will create a permanent separation between the haves and have-nots, more so than income or anything else. People worry that there will be a biological difference, and so far that hasn't materialized. That same year, synthetic insulin became possible, which was a boon to diabetics. Patented bacteria, this is Diamond versus Chakrabarty in 1980, where a genetic engineer developed a bacterium that was able to break down oil. Some bacteria could actually degrade some kinds of oil, but he was able to devise one that could break down most types of crude oil. He thought it would help treat oil spills. And the Supreme Court said, yeah, you can patent organisms that you created. Cool. Shotgun sequencing, genetic amplification. So this is PCR, polymerase chain reaction. Those of you that know what that is know how huge of a deal it is. It's one of the top 3 patents in the US Patent Office that more patents have cited than any other. So it's a big deal, but it's hard to express to people just how much easier it's made life for laboratory scientists, rather than having to spend days of meticulously moving one bit of genetic material from one petri dish to another. You could just basically streamline this. So that's a good thing, but the more interesting one, from the perspective of this particular section of the class, is the notion of a patented mouse, the Harvard Onco mouse, whose patent ran out a decade and a half ago. That was weirdly controversial, right? So they created a mouse that was able to get all of the cancers, which is useful in figuring out how to treat cancers. And the Supreme Court, I guess, because they had ruled on patenting bacteria, this applies there, and lots of countries granted a patent for the Harvard Oncomouse. Then there's a big long period called the Human Genome Project. So I'm going to go into that and break the timeline a little bit. The Human Genome Project lasted a little over a decade, and it took a lot of time. And you have to imagine what it was like before we knew the human genome. We had lots of thoughts about what genetic modification, what genes were, how many genes humans had. We thought we had about 100,000. It turns out we had closer to 25,000. We spent $3 billion trying to look for genes that cause various things. And we took genomes from a bunch of samples from anonymous donors across racial and ethnic groups. Keep in mind, on the eugenics slide when we talked about racial population naturalism, how the, quote, racial and ethnic groups are not obvious to the naked eye. And in fact, it may be that we oversampled some groups rather than others because we were basing this on old, antiquated notions of race. So the Human Genome Project, it returned results, but it's important to know that we didn't figure out the genome, the human genome. The goals were to discover the genes responsible for all human traits, but it turned out that we had far fewer genes than anybody expected. Some we inherited directly from bacteria. Most genes did not behave the way Mendel thought genes behaved. Genes for intelligence, for height, for skin color, for various things just weren't like that. And so it complicated the picture of the human genome. In the 1980s, it was thought that we'd find the gene or genes for intelligence, and now that just seems like a poorly framed question. So that took place over a little over a decade. During that time, we started funding embryonic stem cells when labs that had retrieved lots of embryos or had created lots of embryos from couples had them left over, and they weren't quite doing anything, and the couples didn't want them anymore. And people thought, well, actually, there might be a lot of research we can do on these. So we started funding embryonic stem cell research. But then in the beginning of George W. Bush's presidency in 2001, we decided to limit this research. And the reason for that was because George W. Bush thought that there was moral reasons why not. So there's a video if you click the image here. But the point is that these guidelines recommended limiting the stem cell research to the ones that were already created, and started a medical and ethics council to study the ramifications of biotechnology. There were a lot of funding from the US government at the time, and there were more private funds available. George W. Bush, it was his first veto to ban, to keep this ban in place when Congress tried to lift it in his second term. So what happened? Well, what happened was George W. Bush spent weeks agonizing over this. He brought in ethicists, scientists, religious leaders of various sorts to try to address the question of whether embryos have souls and whether research on them was torture or something. A lot of ethicists were arguing that this could prevent a lot of pain in the future, that these embryos had already been harvested and were going to be thrown away if they weren't used. And so, George W. Bush came to this weird compromise position of limiting the stem cell lines, which was what he thought was going to allow science to move forward and help future generations while still respecting his religious principles of only, I guess limiting damage to 71 cells? I don't know. But I take it seriously. I think he really worried about this. And when you see the video, it's interesting to know how much he cared, which I didn't. I didn't understand at the time. A lot of researchers that were working in this area left the United States to go to other countries, or left these projects to work on other things because funding slowed down. And so Singapore and other nations of the world became the world leaders in embryonic stem cell research. In 2009, we discovered the ability to induce pluripotency into stem cells. What makes stem cells so special is that in embryos, they can develop to be any cell in the body, right? So an embryo is just a single cell, and these could develop. A zygote is a single cell. Embryonic stem cells could develop to become any cell in the body. It turned out that we can take adult stem cells and kind of de-age them and then re-age them into different body cells. But that was only discovered about 8 years later. We still can't do embryonic stem cell research using tax dollars on new lines. There were only about 24 lines that turned out to be effective out of these 71, and a lot of them were genetically homogeneous, which actually really hurt American research into this. So, okay, and we'll keep along with this this stem cell discussion. Induced pluripotency discovered in 2009, of course, as I said. And now there's some approved stem cell therapy. right, so stem cells could become any cells. Okay. We talked about patenting organisms. We talked about patenting genes. What about a gene that naturally exists, but that a company discovered? So in 1994, scientists discovered a gene that was correlated with breast cancer. So if you had these genes, you were many more times likely to have breast cancer than not to develop breast cancer. This is BRCA1 and BRCA2. Myriad Genetics became the owner of this gene and offered a diagnostic that was about $3,000 to check to see if you had these genes, and they wouldn't allow any other companies to do this. That went on until the Supreme Court ruled it invalid in 2012. Myriad Genetics had patented this naturally occurring gene, and the Supreme Court said, well, if anything is naturally occurrent, you didn't create it, you discovered it. And you can't patent America, you can't patent a river you discovered. Why would you be able to patent a gene that you discovered? So the patenting naturally occurring genes was ruled invalid. All right, let's go back in time a little bit. We've a lot of stuff was covered during the Human Genome Project, so we've bounced around in time a little bit. I apologize. Let's go back to 1996, I believe. And who's this? What is this? This is a sheep. A lot of people probably guessed that it was Dolly the sheep. It's not just a random sheep. Why would I bother getting a picture of Dolly, the actual sheep? That was the first animal that was cloned. She died a couple of years later. That was actually a picture of Dolly, by the way. But yeah, we cloned an animal in the mid-90s, and turned out she had arthritis. A lot of people were worried about this, that somehow we'd cloned her from a bad egg, an old egg. But further clones from Dolly's donor cell turned out okay. In 1999, Jesse Gelsinger had a rare disorder that required him to take dozens of pills every day in an extremely low protein diet. And he opted literally as soon as he turned 18, to take part in a project that was going to use genetic modification technology to modify a retrovirus. So retroviruses are viruses like HIV, viruses that insert their genetic code into your DNA. The idea was we would be able to modify the virus so that it wouldn't insert its genetic code, but it would insert the genetics that we wanted. So Jesse Gelsinger was infected with this virus and died shortly after. There were a lot of risk factors that people, through their kindness of their hearts, overlooked because they were so worried about Jesse's plight. Nevertheless, this led to an effective moratorium for the next couple of decades on human genetic therapy. The science just wasn't there yet, and we didn't know how to stop this immune response that led to Jesse's death. In the early 2000s, we developed the HPV vaccine, published the human genome, the HeLa genome, in 2013, which I talked about in an earlier lecture. And then we modified an embryo. And in 2005, this led to this 2015, this was announced in China, and it led to a big international hubbub and the call for a moratorium I was there during one of these events, these meetings, the National Academy of Sciences and other academies of sciences getting together to discuss this. And scientists all around the world agreed that they were going to no longer modify human embryos. Immediately after the moratorium. And then first there was a 3-parent baby, and then there was a baby with a genetically modified, born from, actually many babies born from genetically modified embryos to make them immune to HIV. This was Dr. He's work in China, or at least the baby being modified to be immune to HIV. It's not obvious that is actually the case, that these children who were born a couple of years ago are actually immune to HIV. But he was arrested, and so it's not obvious it was good science. I think he was just trying to get on the history books. This is the 3-parent baby from before. What happened here is the mitochondria from one mother was put into the egg of another mother and inseminated with the sperm of the father. You acquire all of your mitochondria from your mother, and her from her mother, and all the way back basically to mitochondrial Eve. And so some people have mitochondrial disorders. And so these parents did not want their child to have a mitochondrial disorder, so they had mitochondria implanted into the zygote to create a baby that didn't have the disorder that the mother had. And now we wrap up this timeline by pointing out that a lot of this work has led to a thriving industry of private companies that are interested in using genetic technology and genetic modification technology in order to create new therapeutics or enhancements or something of the sort. We'll discuss those in more detail later. The point is that this has reached private industry, and innovation will skyrocket, and I don't think a lot of careful attention to the downsides will happen for quite a while. And we might see some atrocities that we'll be teaching in future bioethics classes. All right. That's it for this video. I'll wrap up this section on the next video. Cheers.